Today we're going to discuss how large-scale changes to chromosome structure and number can result in a clinical disease. The objectives for today's podcast are to review the concept of gene dosage and to define aneuploidy and rearrangements and understand both of their impacts on clinical genetics. Here are the new terms we'll be going over today. Chromosome abnormalities can either be numeric, meaning there are more or less than 46 chromosomes, or structural. They can involve one or more autosome, sex chromosomes, or both. They have a huge clinical impact as they are present in nearly 1% of live births and in up to half of all spontaneous first pregnancy losses. Chromosome abnormalities also account for a large proportion of congenital malformations and intellectual disability, and contribute to both infertility and pathogenesis of cancer. Unlike with single gene disorders, where the cause for the disorder is found at the sequence level, with chromosome disorders we are less concerned with the actual sequence of the gene. Rather, these conditions are a result of quantitative changes to the gene number, like having too many or too few genes. Interestingly, we have learned that some genes display copy number variation, as we have learned, so not all variation in gene number is inappropriate. However, inappropriate gene dosing can nonetheless cause a genetic disorder, depending on the gene in question. We have learned that people have two copies of each chromosome under quote-unquote normal circumstances. However, there are many people who walk this planet with 45 chromosomes or 47 chromosomes. For these individuals, they have either less or more than the expected number of chromosomes. For some, they have no apparent phenotype, and for others, they have a clinical disorder. Another way to describe this phenomenon is by using the term gene dosage, which suggests that under expected circumstances, there is an ideal level of gene expression, and if there is any deviation from that level, a pathogenic phenotype can result. Now, not all nuclear genes are present in two copies. For example, those assigned male at birth will often have one copy of the X chromosome and one copy of the Y chromosome. Furthermore, those individuals with two X chromosomes have their X-linked genes expressed from only one of the chromosomes. In these cases, the ideal gene dosing comes from one gene, not two. Furthermore, those individuals with two X chromosomes will have only one of each of their X-linked genes expressed, as opposed to both of them. We will be learning more about sex chromosome-related clinical disorders in a future podcast. The most common chromosome abnormality is called aneuploidy, which means having an abnormal chromosome number due to an extra chromosome or a missing chromosome. Aneuploidy occurs in at least 5% of pregnancies according to the literature, although the number is likely much higher because aneuploidy is a common cause of first trimester pregnancy loss. Most individuals with aneuploidy either have trisomy, where they have 3 copies of a particular chromosome, or monosomy, where they will have only one representative of a particular chromosome. The most common mechanism for aneuploidy is called non-disjunction, which occurs primarily in meiosis, but it can happen in meiosis too. Hopefully, you recall the process of meiosis and gamete formation, particularly how duplicated chromosomes will line up along the center of the cell and will segregate into new cells. Looking at this schematic, you will see that this happens twice in meiosis. If chromosomes don't segregate properly in meiosis 1 or 2, the resulting gametes may have an abnormal number of chromosomes, which when combined with a normal gamete, result in a zygote that has aneuploidy. If non-disjunction happens in meiosis 1, all the gametes either have too many or too few chromosomes. If it happens in meiosis 2, half the gametes will appropriately have one representative from each chromosome, but the remainder will either have both copies of the chromosome that underwent non-disjunction or none at all. Sometimes, non-disjunction will occur during mitosis, any time after the zygote has been formed. When this happens, the aneuploidy is usually constricted to a particular cell line, and the individual would be considered what we call a mosaic for having some cells with 46 chromosomes and some cells with more or less. This can be clinically significant or have no impact on the phenotype whatsoever. For the most part, aneuploidy is not compatible with human life, which is why chromosome abnormalities are so common in first trimester pregnancy losses. However, there are some forms of aneuploidy that are indeed compatible with life. For example, patients can be monosomy for having one X chromosome, which is known as Turner syndrome. The most common trisomy we see clinically is trisomy 21, where a patient will have 3 copies of chromosome 21. 95% of patients with Down syndrome have trisomy 21. The remaining 5% of patients have a different genetic cause of the syndrome, most often involving a Robertsonian translocation, which we'll talk about shortly. Because it's such a common diagnosis, we'll be talking about Down syndrome in a dedicated podcast later on. The 2 other trisomies that are seen in live-born infants are trisomy 13 and 18. Along with trisomy 21, these are the 3 trisomies that pregnant people can choose to screen for in pregnancy. As I mentioned earlier, abnormalities in chromosome structure can also result in a clinical phenotype. Structural abnormalities are less common than aneuploidy, affecting approximately 1 in 350 births. Just like aneuploidy, they can be inherited through the germline, or they can be a result of a somatic mutation resulting in mosaicism. Structural rearrangements of a chromosome, like when part of a chromosome inverts itself, or when 2 different chromosomes swap sections, can either be unbalanced or balanced. An unbalanced structural rearrangement is when there is missing or extra chromosomal material, resulting in a gene dosage disruption and a possible clinical phenotype. These types of rearrangements affect about 1 in 1,600 live births. Unbalanced rearrangements are often due to deletion or duplication of multiple genes, resulting in a partial monosomy or trisomy for a collection of genes. There are also unbalanced rearrangements due to ring chromosomes and isochromosomes. A balanced structural rearrangement is when all the necessary genetic content is present, just not in the right place. Most balanced structural rearrangements will have 46 chromosomes. Balanced rearrangements are common in as many as 1 in 500 people, and usually don't result in a phenotype because all the genetic information is accounted for. Balanced rearrangements can be due either to inversions or translocations. An inversion occurs when a single chromosome has 2 breaks and then is reformed with the broken segment, but the segment is inverted. Translocations occur when segments of different chromosomes are rearranged between each other. Some translocations are reciprocal, meaning that the total number of chromosomes and their genetic content remains unchanged. There are also some translocations that are not reciprocal. The Robertsonian translocation is the most common type of chromosome rearrangement in humans. These translocations occur when chromosomes whose centromeres, which is usually in a central spot on the chromosome, are near the end. Interestingly, the steps that create a Robertsonian translocation result in a loss of the short arm of the affected chromosomes, and the resulting karyotype is 45 chromosomes. Despite having monosomy for a total chromosome number and a change in the gene dosage, these translocations are considered balanced because the genetic material that is lost really isn't all that important. Robertsonian translocations occur most commonly between chromosomes 13 and 14. Individuals with balanced rearrangements, while not having a clinical phenotype themselves, can produce gametes with unbalanced rearrangements, resulting in a genetic disorder. Balanced translocations are more common in couples who have had 2 or more spontaneous pregnancy losses and in males who are infertile. When reflecting on chromosome disorders, I pause when I think about the medical community's use of the words normal, abnormal, disability, and so on. What the medical community and society has deemed to be abnormal may indeed have health consequences. What falls into that category is socially constructed. I think about how I was born with terrible eyes. Without glasses or contact lenses, I was unable to see clearly more than about 3 inches in front of my face. At a time or place where glasses or contact lenses were not readily available, this would have been a pretty significant disability, I would think. But our world adapted to make it so that this disadvantage that I had was not a disability at all, because we developed the technology so that I could see perfectly with glasses and contacts, and then I got laser eye surgery and can now see clearly all the time. Think about this when communicating about genetic disorders, disability, abnormal, and normal with your colleagues and with your patients.